Good afternoon, everyone. Today's our topic is amino acid and protein. Already in the previous classes, we have learned regarding the protein only. But somehow, if somewhere the question asks regarding that write about the method of preparation and the chemical properties of protein, then what will you write? At that time, you have to write regarding the amino acid only. So, what is amino acid? So firstly, we should know about this one protein. What is protein? So your body cannot the function without proteins, and in order to build all these important proteins, your body needs a amino acid. So in this particularly today's topic, we will learn regarding the amino acid, that what is amino acid and how they look like. So human body has a thousands of a different proteins, and all of which are necessary for the staying alive and healthy. Now what is the meaning of this particularly slide? So as you all know that there are 26 letters in our alphabet, and all those letters are linked in a specific way to build a words. By just changing one letter, you can make a new word with an entirely new meaning. For example, some of you like to eat this one dessert or this dessert, because these two words differ in just one letter but have a different meaning. We eat a yummy dessert, not a dessert, which is a very hot and sandy place. So these 20 amino acids of our human body are like the letters of the alphabet only. These amino acids are the building blocks of protein. As you can see, this one is our, suppose that if a protein is your this particularly building, then amino acid will be block. It means when amino acid come together like a bunch of a small blocks to form a large structure like protein, it's simply like that. Okay, so amino acid are linked together through the peptide linkage to make this particularly our protein. Now just see, these are two amino acids is there, and two amino acids are linked with each other. With the removal of this particularly water, you can make like this type of amide linkage. Okay, so this one is called as peptide bond formation. So now all these enzymes in our body, this particularly certain types of protein is also called as enzyme, which help the speed up biochemical reaction in our body. And without these enzymes, we simply wouldn't be able to exist. These enzymes and other proteins help our body grow and repair itself in general. In our hair also, there is a keratin protein present. As I had told you that, as I told you in the previous classes regarding the keratin, that this keratin is, it is found in our hair, skin also having a protein, and the nails also. And I have also told you one example of a rope, that if you think of a thick rope which consists of a very different and smaller pieces of the rope, then particularly this particular hair is also a protein. Now the protein rich diet, these are our diets through which we can make up our body. Amino acid having a two groups is there, amino group and carboxylic acid group. Amino group, which is a basic in nature, carboxylic acid, this one is acidic in nature. This in this slide you can see this one is amino group, this one is carboxylic acid group. Now this one is the classification of amino group, and now this one is the classification of amino acid based upon the amino group. Whenever, now just see, as I told you in the previous classes, the carbon which is directly attached to this particularly carboxylic acid is called as alpha carbon atom. Now just side to this one alpha are called as beta, and this one is gamma. So when amino group is found on the alpha carbon atom, that one is called as alpha amino acid. Beta amino acid, when the amino acid or when this one amino group will be found on the beta carbon atom, that one is called as beta amino acid. When amino group is found on the gamma carbon atom, is called as gamma amino acid. Now depending upon the nature of amino acid, neutral, acidic and basic. Neutral means, neutral as the name indicates, everything is neutral is there, means there will be equal amount of amino group and carboxylic acid group. Acidic means when the number of COOH group will be more in compared to NH2 group, as you can see in this particularly example. In this particularly example, you can see There are two cellulose group is present, but one amino group is present. So that's why this one is our acidic amino acid. Basic amino acid means number of NH2 group is more in compared to cellulose group. Essential and non-essential. Essential, as I told you, those amino acids which are essential for our body, but our body cannot make it. That's why we have to take these essential amino acids through our diet only. The example are these things. This one is the trick to learn this particularly essential amino acid. A means arginine, V means valine, H means histidine, I means isoleucine, then leucine, then lysine, then methionine, then phenylalanine, threonine and tryptophan. Okay, threonine and tryptophan. While the non-essential except these AV amputative, whatever amino acid you will see, that all other amino acid is called as non-essential amino acid. Now this one is the method of preparation of amino acid. This one is the method of preparation of amino acid, but if in exam the question asks write about the method of preparation of protein, there also you have to write these type of synthesis only. The first one is from alpha halogenated acid. Firstly, you have to make a one table where you will write this heading. On that basis of the heading, you will attempt all those things. Firstly, from the alpha halogenated acid, as the name indicates, on the alpha carbon atom, there will be halogen. So that's why this one is called as alpha halogenated acid. So this on the this one CH3, CHCl, COOH. When it will react with two molecules ammonia, it will be converted into this particularly amino acid, and ammonium chloride will be eliminated. When you will take your one rough copy, then only you will just observe that how this one ammonia it is going to be react and how it is going to be converted into this one ammonium chloride. The next one is Gabriel phthalimide synthesis. This one is also called as a naming reaction also. So firstly you have to learn this particularly potassium phthalimide, and it will just react with this particularly compound. What will be happen? KCl will be eliminated. You will get N CH2 COO C2H5. When this particularly intermediate compound it will be hydrolyzed, three molecules of water will be added. What will be happen? As you can see, HOH, HOH and HOH. So firstly from here C6H5 COOH, this acid COOH, it is eliminated. You will get a NH2 CH2 COOH amino acid. The next one here, ethanol will be eliminated. Next one is Cook synthesis. The starting material is pyruvic acid. When it is going to react with NH2S hydroxylamine, then you will get oxime. As I told you, whenever there will be addition with the NH2S, then always there will be double bond NaOH. It will be added. The next thing, whenever this it will be reduced, what will be happen? There will be removal of oxygen, and you will get this particularly amino group. The reaction of Hofmann degradation, bromine plus KOH. Now from here, when you will take a two molecules of KOH, so 2 kBr, potassium bromide will be eliminated, carbon dioxide gas will be eliminated. You will get this particularly amino. When you will hydrolyze HOH, so one side H, one side OH, so COOH, RCH, COOH, NH2, and C2H5OH, ethanol will be eliminated. This one is a chemical properties due to COOH group. Wherever you will see that all these particularly reactions are only attacking on this particularly COOH group only. Carbon dioxide will be removed, you will get this particular compound. When it will be reduced, then what will be happen? As I told you, aldehyde alcohol will be oxidized to convert aldehyde and ketone. Aldehyde ketone, it will be oxidized to convert into an acid only. So similarly here, the vice versa, when acid will be reduced, it will be converted into a aldehyde ketone. The aldehyde ketone, it will be reduced into alcohol. So there will be two times reduction takes place. So it will be converted into NH2CHR CH2OH. And the reaction with ammonia, what will be happen now from here? This one HOH, water will be eliminated. You will get a CONH2. With NaOH, there will be sodium salt will be formed, and this one HNOH, water will be eliminated. Reaction with the amino group, whenever it will react with this particularly HCl, what will be happen? It will make a salt and H3Cl. Now this compound, when it will react with this acetyl chloride, it will make a compound like this, NHCOCH3. H and Cl, it is just eliminated. Again NHCO CCH5. Now from here this hydrogen, this one chlorine, HCl is eliminated. Now NOCl, nitrosyl chloride. Again from here what will be happen? This one nitrogen and this one nitrogen, two nitrogen It means nitrogen gas will be eliminated, H2 and this one O, oxygen, water will be eliminated, and you will get this particularly compound. This one is due to both amino and COOH group. Whenever you will heat this particular compound, what will be happen? NH2, and from this CH2 one hydrogen and H3 gas, it will be eliminate ammonia. It will be eliminate. You will get CH2 double bond CHCOOH to maintain its valency. That name of this compound is acrylic acid. Next one is Dakin-West reaction. You can react with the acetic anhydride also or acetyl chloride also. This one is a acetylation reaction. Reaction with phenyl isocyanate, CCH5 NCO. As I told you, isocyanate you can represent with NC, and cyanide you can represent with a CN. And this one is also called as a Haydn-Twine formation. It will make a RCHCOOH and H. One hydrogen is left, so one hydrogen it will move to this side. So NHCO, NHCCH5. This one is the main role of our protein, that how these proteins are used in our body in the form of particular DNA, antibodies, hemoglobin, hormone, digestive system. It means in the form of enzymes. So these things. Thank you.